I welcome all the participants again for this session of Speakers Roundtable. In addition to our existing speakers whom you already met, we have now two more speakers in this session. I have privilege of welcoming two great leaders from Indian industry for this forum. First of all, I wish to welcome Mr. Ashwin Upasne. Ashwin is a highly mature pharmaceutical professional with a great sense of humility as a leader. He is currently leading Cipla's Goa operations as a head of technical operations. He is a postgraduate in pharmaceutical operations and management. In his past 25 years of experience, primarily split in operations, quality, and project management, most of his career has been exposed to manufacturing of sterile products and aseptic manufacturing. Welcome, Ashwin. Now we have another stalwart from the industry, Mr. Chetan Pathak. Chetan is serving as a director of quality with MCure Pharmaceuticals. Chetan is a master of science in microbiology with 25 years of experience in QC, QA, microbiology, validations, and corporate quality within a pharmaceutical manufacturing environment. As a site QA head, he led multiple successful regulatory inspections like US FDA, MHRA, TGA, ANVISA, WHO, and many other sterile and non-sterile API, oral formulations, and injectables. He has also experience of leading corporate vendor qualification, vendor audits, QMS, and API contract manufacturing teams in regulated environment. Let us welcome Chetan to this session. Welcome, Chetan. With this, I'm ready to begin my speakers roundtable. Now, let me start with some of the questions from yesterday, which we left, we couldn't attend. I have a question for Vipul. Vipul, there is a question that US FDA is in discussion for 2 to 4 metric, where you are suggesting lot many. Why is it so?
Can you repeat the question, please?
So question is, US FDA is in discussion for 2 to 4 metric, whereas you are suggesting lot many. What is the reason?
Look, one thing is people have to be very clear that US FDA is not asking only 3. They are, because this discussion is happening from last 5 years. Initially, they came with a set of metric, which includes even the very difficult one, kind of a quality culture they want to monitor. Then, but industry continuously discussing, and in another thing is that and they actually want to start this activity. So, in a new revision of this guideline, they have very clearly said that start with 3 for reporting, but others you should not be limiting yourself to that. You should be having as many numbers as you want to cover your risk area, so that you can have a full understanding of your gaps. If you are aware of gaps, then only you can work upon action. And why FDA want this metric? Basically, FDA want to cover 2 things. One thing which is very important is they want more data to analyze to cover the risk of inspections. Second thing is they are also want to proactively deal with the shortage of drugs. So if they have the product based metrics what they are talking about, they will be having well informed or information about the product, how the products are behaving, and whether this product is going to have problem, and all predictive kind of analysis they can do. And they can take care by informing companies or coming to the companies for inspection and understanding whether this product will be there for a market or not. So it is not that FDA is going for 2, 3. It is only just for starting a reporting system.
Good. Then we have another question. How much data you feel sufficient to derive standard targets to start with?
Look, to have a simple answer, I should have a data 3 months to 6 months continuously monitor data. It is not that 15 days in a month and then another 15 days of another month. No, it is a continuous data. What is generated 3 months to 6 months will be good enough. And it is not only the data. When you set a target, you also have to talk to your management. So companies require to understand what management is looking at it. I can give you just one example that if a company is looking that as soon as material comes, it should be available as a release. Company can ask. They want zero inventory kind of a planning system. So if a company is receiving 50 lots or manufacturing 50 lots a day, then and if you expect QA and QC to be as soon as samples comes, you will test and release within 8 hours. It is possible. It is not possible. Everything is possible in the world, but it will require resources, manpower also, as well as instrument support also, and other infrastructure also. So you have to balance out that cost with the management, what you require, what is feasible. So it should not be coming up that return of investment will not be coming to you and you will be coming in losses because you don't require a pending sample. So it is all rational and logical thinking what we have to use when we are setting up a target.
Great. So Chetan, would you like to add something on this?
I think last point which he raised, if we need resources, it adds cost. So it should not happen that to get faster release, we add on to the cost. That's the point. I'm fully in agreement with that. That's the right thing, I think. That's all from my side.
Good. So now I come to Alvaro. So with the question of which disinfectant should I use? I mean, there were questions, several, placed in different forms, but it's ultimately indicating the disinfectant rotation. So which disinfectant should I use? How frequently should I use sporicidal agent? So this is a different form of the questions, but ultimately it is pointing towards the rotation of disinfectants. Can you give some throw some light on this, please?
Yeah, you can hear me now. Okay, so where do we start to answer this question? The first is to look at EM data. What sort of vegetative microorganisms you're finding as a routine in your clean rooms, and which ones are you getting a few times per year, the odd cases. With that in mind, then you go to what we advise as the broad spectrum disinfectant that cover most of those microorganisms. And which one would you use? There's many molecules available, quaternary ammoniums that are high in terms of detergency, phenols that are very effective. The advice is to select the one that kills as much possible, but with a lower residue limit. That's why normally quaternary ammoniums are widely used. They are commercially available from many, many vendors. Once you have the base molecule that covers broad spectrum, you need to include the sporicidal. For that, you really need to base on the EM data, the worst cases you have. And the frequency, at least you should start with a weekly or bi-weekly base. Gather data in practice to see if your EM keeps safe or not. For example, you decide I'm going to start my rotation doing a bi-weekly rotation of the sporicidal agent. It's not confusing to operators if I ask them to do it every 2 weeks. So I go every 2 weeks. And my EM data show that I get sometimes spore contaminations. So what do I need to do? I need to go for a weekly application. What if I'm still getting spores? Instead of going to a more frequent use than a weekly, you should ask yourself what I am getting these spores coming into. Where are these spore-forming bacteria coming from? Are they operator source? Are they coming through trolleys? Is my disinfection of materials not being good? We should try to stick to a weekly at the most use of sporicidal agent. It's not it's not a very good idea to go for a daily use of a sporicidal agent. We have seen this done in many companies worldwide that they go for overkill with a sporicidal agent. First of all, that shows a lack of trust in the system itself. Second, it may be highlighting that you have some problems with vectors of contamination. And third, you are putting a lot of pressure in terms of the deterioration of your facilities. So keep 1 week, once every 2 weeks as a reference. Sporicidals, go for the molecules that are faster killing. These are the most critical ones, the ones that achieve a faster killing effect. And second, go for the ones that leave lower residues possible. And finally, include rinsing action or cleaning action of residues. So a sterile detergent is a sound practice for your facility. It will keep your facility running smoothly all year round. It will protect your investment in capital terms, and it will also make the activity of the biocide more effective. I hope this roughly clears what a rotation scheme will do. The only last aspect, I mentioned that before in presentation, is for equipment itself, you may need to use something, hydrogen peroxide, either BHP, if you have the technology available for the machine, or for some parts, you may need to do proper wiping with 6% hydrogen peroxide. And I would advise a microfiber for that fashion because it increases the activity. So that will be a fairly robust, also including obviously alcohol as a routine-based product. I hope this roughly clears the situation.
I think need for very conscious applying the sporicidal agents, because on one hand it is helpful, it is required, but on the other hand they are very much corrosive and it can damage your machines and equipment surfaces. Yeah. There's another question I would put it for Ashwin. So the question is, we produce liquid injectables like NS and DNS for European market. Do you think that we should consider implementation of PubSit for this?
Yes, definitely. Why not? New requirement of Annex 1, as well as there are certain expectations from the inspector that we should have PubSit for all products, whichever we are manufacturing, for at least for European market. And PubSit will give you, it's not only the, it will give you assurance about the filter readiness for the use. And that's how I would personally prefer to use PubSit for all products rather than only focusing on some specific products.
Yes, yeah, I support that. Just one comment, unless and until you have, because of your process flow or your system design, you are posing some additional risk to your system by implementing PubSit. However, there are today lots of systems available, ready-made systems available, which can allow easy implementation of PubSit. So, but I would recommend all of you to go into PDA paper. There was a very good publication in PDA later about PubSit. So they did a study on where are the risks actually for implementing PubSit and how can you do that. So I would recommend all of you to go to that PDA later, which talks very well about the PubSit. So there is a question for Taimur. It says that MES validation, I think question is not very clear to me, but I will try to read out. So MES validation and requirements traceability metrics. Can you say a few words about? I think he wants to know about how can you do this? So,
I mean, let me take it in two ways. I mean, what my understanding is, MES validation is one question, and then requirement traceability matrix is another question. I will treat as such. I mean, MES validation is like MES is by itself category 4 system that's highly configurable, right? And then in that system, there is a one configuration aspect that needs to be taken care of. And then also in the MES systems, if there are interfaces back to equipment or back to sometimes weighing systems, right? So if MES is collecting the information outer systems, so this validation of interfaces in the MES becomes very, very crucial. And then design of the interfaces becomes very crucial. And I think out of all the products in the MES market for pharma, I mean, one should choose the product who has much more standard interface. Because when you go again and then when you begin to talk to people, they will say from the boys from the street, PLC boys will tell you they can connect any machine for very cheap price. But the answer is, of course they can do it again, whether it's function or not. Here for the MES, I mean, this notion of interfaces between the shop floor and also between the upper systems like ERP and bringing the information back and forth, ERP system becomes utmost crucial areas. And then the rest of the MES system by itself is, when you use it for electronic batch recording, basically a paper-based form which is created into the system and flowing through the approvals. So this section, yes, there is a design issues, there are some logic behind it, but they can be handled much more smoothly. But when it comes to these interfaces between upper system and shop floor, so I think you should be putting much more attention to that one. And then again, my experience goes that more and more MES is coming to the shop floor systems, then more and more there is a risk. And then, I would push those implementation further ahead. Let the operators enter the information manual, right, at the beginning, because your maturity won't be there. And then most likely, you are not only mature with your mindset, but technological maturity is not there in your area, because that's also another maturity level. So I would say for the MES, and then, the ERP systems, especially SAP is the big name in pharma, between any MES system is very well defined usually. So you can find a lot of resource and knowledge around that. You have to do it. So do that one, and then try to do it MES. If you want to come to the shop floor, mainly limit as much as possible with the weighing systems. So because over there are standard interface and knowledge built over there. So that's, I would say, for the MES system. And then requirement traceability is, utmost important document that needs to be created, that should be managed during the process. And then requirement traceability is over there not only proof that my requirements are meeting my expectation through the test specification and design, but actually requirement traceability is there for you if you're going to do later on any upgrade to the system. Then you should come to the requirement traceability matrix, do your assessment, and then make, based on this requirement traceability, what level of regression testing you will do later on when you come to a major upgrades. So our experience shows, again, if you go for only the validation as a documentation, traceability matrix is created once and becomes part of the validation report, sits there forever, because there is no culture into the company and understanding to update it and to use it. So it stays there. When you do the upgrade, then you don't know what the scope of the testing would be for the regression. Then you fail. Very simple. You fail. Not only you, big banks failed. Not only you. Who are you as a pharmaceutical company? The big banks failed. The big airlines failed. Lufthansa failed. Royal Bank of Canada failed. 1 week, Royal Bank of Canada cannot receive money and send money. So they failed. Why? because they don't maintain this information. They don't know what to do. And then more and more we will be coming with CSA stuff, and then more and more automatic testing tools will come out in our life. Then this requirement traceability will be the key again for this validation activities, computerized validation activities. And I think, again, if you say me, Timur, what value you add out of 30 years in this business to your clients, I would say designing the requirement traceability matrix, maintenance, and then, teaching the people how to use it later on in their applications. So that's from my end. So, Brett, up to you. Subhata, you are mute.
So I add from this point of view, because I had experienced this in the past. So to implement your hard copy of the document in toto into MES, you are going to fail drastically. So there is a need for mindset change. It is altogether a different ball game. So that's what I can say, with the experience of using MES in my past. So our next question is, I would ask this to Vipul, as well as maybe Chetan can add. How does parametric release work? What are the regulatory expectations related to parametric release?
Chetan, you want to start?
You can start. I will join later.
Fine. Look, this again, like in your presentation, Subrata, you were talking about rapid microbiology, and parametric release also still is a very challenging thing for industry to make it a routine thing. Regulators are asking parametric release can happen only on special cases where where drug is not very stable, or where the need of a drug is so critical that it will have a patient implication, means it will not reach to the patient, then like something condition like pandemic. So those are the things when they allow. Still validation for parametric is not very well formed, so that people are not, or regulators are not assured enough for parametric release. So it is started in somewhat way, and it is quite possible. It totally depend upon what we should do, and very two very I think Temur as well as Alvaro can add to this, how to make our system of operation robust enough so that we can have assurance for quality people itself, as well as the regulator will get assurance of a product which is released on a parametric basis is good quality and it will not harm the patient. This is my view on this, Subrata.
Chetan, you would like to add something?
I think parametric release expects two, three things. One is the control, process control. Second thing is process understanding. And that's where whether our process understanding is robust enough, we are confident enough on our own processes, our own understanding, whether it is adequate. These are answer to ourselves first, because to achieve that confidence is the first step. So first we need, as said by Mr. Vipul, yes, that's possible. Regulatory has supporting approvals on that, provided that your process understanding, your process control strategy is robust and you are able to prove it to the regulatory agency. I think that's all from my side.
Just like to add my views as well. So I think we need to go more and more, or rather go from dependence on people to automation. When you can establish a system which is self-dependent, self-corrected, and kind of a predictive kind of situation, then I think we are more confident to go for a parametric kind of release. So that's my viewpoint to it. Maybe Alvaro or Timur can add if you feel.
Before Alvaro, you can start, or Temur, you can start. Subrata, I have to say that yes, automation, But automation brings us more challenges, as Taimur says, that when a category of automation becomes category 4 or 5, and assurance you have to build not only at a time of validation, but the assurance required in the life cycle of that particular thing. So that what is always a challenge. You can understand getting one count in class A area, how much difficult life we make in operation. We are still fighting for weeks and months. Sometime we're doing investigation, and finally we decide not to release the product on a safer side. Correct. So again, that why this happens when everything is validated, full assurance is there on the system. Why we cannot have a rational for one count in class A area? Like yesterday, she was also talking about microbiology OS discussion, and she very clearly told, yes, we can take a decision, but it is very difficult to take that decision. So yes, now you can add to it, Taimur or Alvaro, your viewpoint on this.
If you allow me, Tamur. Well, I would, as you both said, especially Chitan, that process understanding it's critical when you venture into parametric release, and the control strategy. Remember that requires the full bubble diagram that I show is not only the filling or the chemical process or the mechanical process involved in the filling operation, but also all the supporting operations in terms of contamination control. So, as you rightly say, people sometimes an investigation can take months, when actually preventing that excursion is probably going to take only a few brave decisions into preventive instead of solving problems. Also, I am as an engineer, I was very shocked the very first time I heard a well-reputed consultant saying that the EM data we get through settle plates or to product plates are only 30% effective, meaning out of 10 positives, we only see 3 of them. We get 7 false ones. So really, the data that we rely so much on and that we can get as complex as possible in terms of data analysis and software, but at the very end, don't lose the sight that this is a very limited tool of acquiring data. So if we want to produce a safe drug, it's really all about the process. And obviously, we need to go into the automation and protection of the process, but we cannot lose the sight of what the reality we live in. I saw a presentation by PDA 2 years ago where they said clearly that isolators are only available to 20% of the injectable doses that are being used in the world today. So we cannot lose our mind frame that people wiping, mopping, gowning, training, and the old-fashioned conscious behavior are the way to protect patients and the people. And we may get too much carried away in the beauty of engineering, forgetting how important is just the day-to-day activity that our operators do. So bear that in mind. All of those technologies are coming to stay, but we live in a different world today, and meticulous and precise thinking process from the design of a good SOP are critical, and then train your people. That's the best way to be safe and robust.
Okay, thank you, Alvaro. So my next question is to
Subhata.
Ashwin. No?
Subhata,
Yeah, please.
Can I add only one small thing, right?
Sorry. Please go ahead.
Yeah, I mean, in general, it's not my field, like let me tell you, but I can tell you only one good news, which is definitely will help or still helping maybe without undoing, like this serialization, like this 2D barcoding, which is on the individual products. When you do a parametric release, that's what I know, because my understanding is you ship the product, right, without it is releasing, right, in the real release, and you wait for the microbiological stuff which is coming, right? But with serialization, at least you know exactly who take this product and then where this product is. If there is any problem happens with the parametric release, right, after 14 days, You have time to find in the pharmacies, and then you can immediately block that product for selling. You see, that serialization may help. It come to my mind right now with your discussion for the parametric release for sure, because then you have all the good reasons, right? On the time the product is on the pharmacy, still you can block it, the pharmacy on that level. So it's a good thing. Still, it doesn't go to the patient. So that's I want to add 2 cents.
I would echo here with Alvaro that we should have it's all dependent on how confident we are on our processes and the sterility assurance level. And for that, we need to have end-to-end process understanding. And if that understanding, we demonstrate that understanding, automatically we can go for the parametric releases.
Correct. So I move to the next question. The question is, Ashwin, so looking at the current scenario where aseptic manufacturing is still largely dependent on people to do major part of the work, and looking at the scenario of attrition in India, what would you recommend? Generally we find that there's a lot of issue with regard to getting the people in, training, retraining them, and then they leave, and again we are back to square one. So what is your recommendation? How, maybe Ashwin, you can start, and then maybe Chetan also can add.
So Subrata, you're very correct that important element in our current operations is the person or our operator. Because today we are very highly dependent on the operator, because some of our processes are not that automated. And then we have to rely on conventional filling machines and conventional aseptic operations. And for that, the operator becomes, and training of operator is a very important thing. I would say it should start top down, that first we should lay down what is the expectation from a management from an employee. And if we set that culture that this is what is expected from them, and we start bombarding those through training, I would say not a conventional training, but demonstrating them what is the requirement, what should the expectation from their side. Secondly, today what we're lacking is the buddy concept. I think we should have tagged new employee with the old employee, and if we do that, automatically the good things which are happening from a experienced employee would transfer to a new employee. If you do that, and automatically, if while training, if we have a course corrected as by while putting them on a job, whether we have some advanced training to them, covering all practical aspects of right from basic microbiology to a operation, maybe an operation for lyophilizer, which is the most critical at this point. If we do that and demonstrate them and allow them to absorb the technology, I think that would help us to make the things better.
For sure. Chetan,
I have some different thing to say on this. Like, for example, if there are like job rotations, like if a vial machine operator is also trained in the filling machine operations, filling machine operator is also trained in the sterilization. So if there are multiple people doing the same activities, if that's the possibility, then yes, we have the backup manpower available whenever resources are less. That is one thing. Second thing, I will come to Mr. Vipul's previous point of cost of resources. Somebody can say that we can put more manpower as backup. If that condition happens, we have some 10, 15, 20% additional manpower available trained on the filling machines or that. But that's a cost again. So as Mr. Vipul said, resources and its cost, if management agrees, then that's another point. So these are the two things, but I would prefer my first point, that is rotation of the job. It should not happen that if the filling machine operator for 5-7 years he is doing only working on filling machines and he is not experienced on while washing machines, or the vice versa. So that is something can be thought of. That's my point on this.
Okay, thank you. So now I have a question for Alvaro. The question is, sorry. Yeah. So what should be the frequency of aerial disinfection?
That is a very good question and very common. So first we need to ask, what is the aim of aerial disinfection? So the aim of aerial disinfection should be to reach difficult areas where manual disinfection is not possible. Then we need to analyze the limitations of aerial disinfection. To properly use and validate an aerial disinfection, you should have the possibility of shutting down the ventilation systems to avoid the biocide getting into the HVAC conducting and creating corrosion issues in the ducting itself or in the HVAC machines. So if your installation is not prepared for that automatic close and shutdown of the ventilation while you perform your HVAC, your fogging, then you probably need to stick to that only for when you open and declassify the zone, when you do have an out of control situation, a trend that is going out of control to try to recover. But you need to address the risk you are creating, either by closing manually the filters or assuming that you need to inspect the air ducting and find out if there's any damage to that. So even though it may sound as a very practical tool to use, to be able to do it in a safe way for installation and in a reproducible and trustable way, it's something that we do not favor much to recommend. Most of the areas are properly designed in a way that you can access them and clean them manually properly. So it's not required unless the structure of the machine itself is intricated and complex. Different story is for the wrap or the isolator or the filling area itself, where BHP technologies and alike are widely used and are the best way to guarantee the machine is sterile. So my answer referred to background and not to the filling areas where BHP is the favorite technology. I don't know if that is clear enough.
I think so.
Subrat, can I add something?
Yeah,
Subrat, I just wanted to add something here.
Yeah, please.
Should I proceed?
Yeah.
Alvaro, I fully agree that about your point on aerial disinfection. If we are disinfecting surfaces well, if persons are taking all precautions, there is no count of air of its own in such critical areas. But the cases which I have seen is the most difficult areas to disinfect are the ducts only. If, for example, if there is a spore contamination observed inside the area, there is always a doubt whether that spore is having habitat somewhere in some corner or even in the ducts. So, and these ducts are unreachable. We cannot disinfect them by reaching there. So that in that case, is it not feasible to have aerial disinfection to treat the ducts? Although your point is well taken that it should not damage the duct MOC, we should select the disinfectant accordingly. But I would go for some frequency to sanitize these unreachable areas like ducts rather than the core zone, because core zone is always under control if you sanitize the surfaces. Alvaro, that's my opinion. Alvaro can say something whether what I'm saying is true or not.
I would just like to add one point here. So basically, aerial disinfection, as long as it is not in the critical area, maybe, the point which you raised, Chetan, difficult to reach kind of situation. So it should not be definitely in grade A area, and I would try to avoid this in grade B as well. And as long as it is outside grade A and B, I don't mind doing it at a some frequency to keep the bioload in control. But Always the step before disinfection is cleaning. If you are not able to clean it, disinfectants doesn't work. So that's my view on this. Yeah, Alvaro, you wanted to add something?
Just to add on your comment and to Chitan advice on the HVAC system. We would consider that as a non-routine practice. So if you do have events and you do that with a proper, perhaps 6% hydrogen peroxide a few times per year, it may not be that damaging to your ducting system. But we need to understand that a lot of times there will be condensation of that vapor, of that fog within the ducting system itself. And those condensation spots are likely to create corrosion issues, which will impair the subsequent disinfections and will also increase the likelihood of microorganisms to proliferate. So it's something very, very difficult to balance. I would suggest to see if it's possible to have visual inspections, or if you have handholds in the ducting system, there are remote swabbing tools you can use for visual inspection, just conducting some swabbing in the ducting system and find out if you do find real evidence that what you're getting in your AV areas is coming from the ducting and not from the corridors. Most of the times we see that the evidence shows is that what you do get in AV areas are coming through people or through materials most of the time. So yeah, you can use it. Just bear in mind that there's such a risk. And if you can build a new facility prepared to withstand BHP fogging through the whole ventilation system, then you are prepared for the next 20 years without problems. But that is a new facility. Existing facilities is a risky operation in my view, but it's a valid thing to do. Absolutely. Thank you.
Yeah. There is a next question which I would pose it for Vipul, Chetan, and Alvaro. I think any of you can pick it, and maybe alternatively, all of you can pick it. What should be the quality release approach for pre-sterile items like wipes, disinfectants, etc.?
I think,
yeah.
Okay,
Look, like everything, what is required release for Israel, it is also a component which is required to be used in the Israel operation or manufacturing operation. So there should be approved protocol or a specification with them that has to be there. And what we should be including it, yes, of course, whether if it is, it all depends upon if you are buying pre-sterilized material or you are sterilizing it inside your factory. And the method of sterilization, all those things required to be explained, and we have to follow this protocol as we are following for other materials. Yes, Chetan, you can add to it.
I think release has to be there. When we say materials manufacturing are to be released, people always, I have seen, they misunderstand it. Something needs to be done. I believe there is no testing required unless there is any parameter. For in disinfectant, what it's and you need to keep watch on your active ingredients and composition, whether active ingredient percentage by the manufacturer. So you need to the composition is correct as per the predetermined specification or validated disinfectant composition. That's the critical thing, and it's not needed that you test some parameter of disinfectant and then release. It's only the comparison of the on the level composition is one example. Likewise, for every atom, what is critical to be checked at the time of release is important, and not necessarily any testing workload need not be done. So, but yes, that's comes in product contact or core zones. There is a specially expectation from European authorities or European guidelines that we need to have specification and release of such material. Specifically, the specification needed, look into it, what we yes, so that is required, but should not be misunderstood with doing some testing for the release.
Just to add what, sorry, should I?
Yes.
As Chitan said, for biocides, for example, the critical things to guarantee that the process of manufacturing is a validated process with change control. So whether you manufacture in-house your own mixes and filter them and introduce them into the AB area with a validated process, or where you buy a ready-to-use product from a vendor, at the end, you're checking the same parameters, either in-house or outside. Outside, you have the advantage that this vendor does that for many clients, so the cost it really is smaller and the risk is smaller than when you do your own validation. Then you need to validate also and include in your release protocol for how long the product itself can be used, whether we're talking about sterile wipes or sterile disinfectant. After I introduce it, for how long is it valid? And you have to establish your own criteria. And then after that, you need to guarantee that the parameters in the certificate of analysis or in the certificate of compliance are checked by your quality department before you actually accept the product. So most of those products will come with individual per lot certificates to guarantee all of the requirements that Chitan mentioned.
Ashwin, you wanted to say something?
Yeah, I just wanted to add two. It's totally dependent on how strong our vendor selection program is, and secondly, how
Agreed.
robust the
Yes.
vendor qualification process is. Because unfortunately, today we are focusing on only on APIs vendor selection and the excipient vendor selection as a pharmaceutical company. But we need to focus on these ancillary items which we have procured for and which are basically utilizing in our trial areas to build stability in our products. So I think if these two processes are very strong, based on that assessment, we should decide what should be our frequency, what should be our release approach for the pre-sterilized material.
Very
But
rightly
definitely
said.
that should have, but definitely we should have a tri-party agreement. If you're procuring for a pre-sterilized items, we should have a tri-party agreement between the company who is manufacturing it, the company who is sterilizing it, and the company who is procuring it. I think that should be very robust.
Well said, Ashwin. And basically, I am in agreement that taking a decision lot to lot, we cannot change that. So everything what is required to establish a vendor and its specification has to be understood by the, and risk has to be evaluated before taking a decision that how many batches we can clear without testing, or what kind of periodic testing is required, or we can depend upon 100% on vendor testing. So yes, I am in agreement with you and Chetan also.
Yeah. There is another interesting question on rapid micro methods. So the question is, how is the regulatory acceptance of it, and what are the critical considerations for validation and regulatory filings? So maybe Chetan, would you like to address this?
I think regulatory authorities have already supported these, and one example is the US GMP guidelines September 2004, which mentions that for microbial identification, genotypic method, I mean, those are recommended. So regulatory agencies are already recommending to use rapid methods like genotypic microbial identification, and genotypic identification is widely used these days by the industry, but other rapid microbiology people are still not using to that extent. But as far as regulatory view is concerned, they are always supporting to this. That is what is indicated. Critical aspect, rather than critical aspect, I would say the PDA technical report as well as USP chapter on how to validate such rapid methods is supporting these decisions. And one thing is which regulatory clear mention, or even that you can always have an alternative method. You need to validate it properly. As far as rapid microbiology, if some company wants to implement that, they can keep on working with regulatory authorities and getting their comments. It's easy for new product to be filed method. So that opportunity should be used whenever there is a new product getting filed, so that regulators, if they have any questions, there will be always question answer things and settle to use permanently in future. So yes, regulatory authorities are supporting all such new technologies.
Okay. So there is another question. I think probably this we should be the last question of this session. So what is the sorry. So the question is, what is the approximate timeline to validate SAP for a large size company? The question goes to Taimur. Taimur, can you address this?
It depends. Usually, the general approach for this one is, validation should not add any single day to the normal system implementation. So if it's well designed, the implementation methodology is well designed, let's say, then the validation should be included in that methodology, implementation life cycle. So that, if the project organization says that, we will implement the system, I don't know, in a year, then validation should begin from the first day of the kickoff, even 1 month before, ideally, so that you prepare validation plan, master plan upfront, and then should finish by the time when the system goes live. So meaning that that you should be adding enough resources for validation, right? Not only like who's going to prepare the documents and all that stuff. And then just one thing is, in multinational companies that we are working with, they don't call a validation consultant. They say project quality managers. So, for example, we are working for a very big multinational company for almost 10 years in SAP rollouts, let me put it that way. And then our title over there is we assure that implementation quality. So our role is project quality manager, not validation guy or validation consultant or senior or principal validation consultant. So, the role is over there. And then my understanding is for a large pharma company, meaning that, I don't know, let's take big Sun Pharma, for example, right? In your case, in India case. If Sun Pharma is there and they have multiple sites, they have in worldwide existence. For this type of implementation, I would say minimum will take 3 to 4 years rollout time. And then our experience with Sandoz, one experience, it took us 7 years worldwide rollout for this implementation of that SAP. And then we begin the project 7 years ago, for example, with a pilot of 300 users. And by the time when we finished the project, there were like 40,000 users on the system, on the single client. That's a big undertaking. But the understanding is here for a mid-size, single implementation company, for a pharma company or experience goes, only one site, it should take between 6 to 9 months. 6 months is very aggressive. 9 months is ideal. When it goes above 9 months, it's a little bit push. Then some people didn't do their job. That's what it means. But that's my experience. Thank you. Subrata, to you.
Thank you, Mr. Taimur. I think we are coming to the end of this 2 days conference to mark the global launch of this company. I would expect any closing comments from all of you.
Thank you very much, Subrata. And this is really a learning experience. All the eminent, I am very thankful to eminent participants as well as speakers, because the questions are also coming up of very good standard, and our speakers are also very knowledgeable, and they are able. I believe that participants are satisfied with the answers. And yes, of course, we are looking for many more this kind of engagements. And to the participant and the other team member, yes, you can understand that we have knowledge available, and we can utilize this knowledge whenever we want. So we all are we we are available to help industry to take it to the next standard. Thank you very much from my side.
Thank you, Vipul. So anyone else would like to come?
I think let me take this opportunity because I joined directly in this panel discussion. Let me take this opportunity to congratulate GxPFONT team, Subrat, Mr. Vipul. It's a great initiative, really a great initiative. And I really enjoyed knowledge sharing today on this platform with all the speakers with me. It was really enjoyable knowledge sharing, and I also got a couple of good new approaches during this discussion. So thanks everyone on this stage. Thank you.
So I would echo Chetan here because even I joined this session itself, but the way questions were came and the way the answers were given to the panelists,
Chetan?
it was a great learning for us also, and it was very good event. I would echo Chetan entirely what he has just mentioned.
Subrata, I mean, 2 cents from me also. I mean, thank you very much. It was organized very professionally,
Sure.
very professionally, and then I know you guys put a lot of effort behind it to make it success.
Right.
And I do think it's a success. And then from our end and from Conval, our team, so we are very, very pleased to connect with you, to know you, and then to know your team. And I think there will be a big contribution from your end also to the Indian and from their worldwide pharma industry. And I'm hoping that we will be working together. And then, if you do any type of presentation like this, and if the audience like it, if you get a good feedback, I'll be more than happy to contribute with my knowledge and share that one together with the group, and also get knowledge also from the group. Thank you very much. I appreciate it a lot.
Thank you, Taimur. I appreciate that. Of course, we would look forward more such opportunities.
I would like to congratulate you, Surata,
So
and,
I thank you.
obviously thank all the participants. It was very interesting to hear from your experience, field experience. And I would also like to address how important these efforts are for the worldwide industry. We are here trying to help patients have safe drugs and businesses to be successful long term. There's two interests in the same table. And the job was perfectly done. I think the people behind the scenes have done a great job in terms of the technology of the platform for hosting this seminar. And congratulations. And we are more than welcome to be invited again if you give us the opportunity. And also bear in mind that here we have our training room also available for some remote training now that the COVID restriction forces us to stay grounded. Let's just carry on the wheel spinning through these webinars. Thank you very much, Surata, and congratulations for the GxPFONT.
Thank you, Alvaro. Thank you, Alvaro. I really appreciate all of you for coming here and presenting. I can understand what it takes to make presentation and come forward and making these presentations. So I really appreciate that. I also at this moment want to thank the Pharma Circuit team, Mr. Kuntal Chatterjee and Ms. Mohua. So for their excellent effort at the background for making this happen. And also I would like to thank my team members from GxPFONT who spent a lot of hard work to really make it happen. And of course, I want to thank all the participants here for taking your time out of the busy schedule and making it to this conference. And we shall also stay connected for future. You can reach out to us anytime. One thing I can tell you, we don't charge for telephonic advice. We would always charge only for, if any concrete solution we provide for your company. We definitely look forward to a greater association for future. Please look at our website and our YouTube channel for more about our company. Thank you.